Hello, everyone, and welcome to today's webinar, Facilitating Transcriptomics of Sensitive Target Cells with Gentle Cell Sorting. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Miltenyi Biotec. To learn more, visit miltenyibiotec.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This presentation is educational and thus offers continuing education credits. Please follow the process to obtain your credits by clicking on the Continuing Education Credits window. I would like to now welcome our speakers, Alina Steinbach, the Product Manager for Flow Sorting at Miltenyi Biotec, and Alexander Jonsson, Research Associate, Department of Immunology, Genetics, and Pathology at Uppsala University. Alina and Alexander, you may now begin your presentation.
Thank you for your kind introduction, and welcome everyone to this webinar. Today we want to discuss how to facilitate transcriptomics of sensitive target cells with gentle cell sorting. The webinar is divided into two parts. In the beginning, I will introduce gentle cell sorting upstream of transcriptomics, and afterwards, Alexander will talk about his exciting research and how he acquired pancreatic microvascular endothelial cells for transcriptomics analysis using the MaxQuantIdo. Let's first discuss why cell sorting is so important prior to sequencing. First, we want to enrich our specific target cell populations and improve the reads. Second, we want to exclude dead cells and debris in order to avoid cell-free DNA and RNA contaminations and decrease the costs. However, cell sorting is coming with some challenges. First, if the cell sorting technique is harsh, dead cells might end up in the sample, and this could increase the costs, and also DNA and RNA contaminations in the sample might result in background. Harshness of the sorting can also lead to gene expression artifacts that lead to false results. Another challenge are timely delays due to slow sample processing, and these could lead to gene expression alterations, again ending in false results of your sequencing experiments. Let's have a look at some exciting data. Unsorted cells and sorted cells have been compared in regards to their background. During this experiment, it was found out that unsorted cells had around 50% of reads actually being from a cell, the rest was due to high background RNA. Sorted cells, on the other hand, had about 97% of reads coming from the cell and only low background RNA, showing how important cell sorting prior to sequencing is. On the upcoming slides, I want to discuss how you can improve your sample quality with using microchip-based gentle cell sorting. The MaxQuantIdo cell sorter is a real benchtop cell sorter due to its low weight and small dimensions. It is equipped with 3 lasers, 8 fluorescent channels, and 2 scatter channels. This provides full experimental flexibility and the possibility of multi-parameter cell sorting. If you are interested in more information, have a look on the website, the image brochure, or the user manual. At the heart of the MaxQuantIdo, there is a microchip. Let's have a look on the cartridge. The cells are inserted into the input chamber. At the bottom, there's a mixing propeller, keeping the cells at suspension at all times. Cells are sorted into the positive collection chamber, and the non-target cells will all flow through to the negative collection chamber. At the bottom of the cartridge there is a microchip. On the right side we see this in more detail. Cells are coming from the input chamber into the channels of the microchip. They are crossing the laser beams, and if the system recognizes a target cell, a pulse will open the valve and the cells will go to the positive collection chamber. All the non-target cells will flow through to the negative collection chamber. Let's have a look at this in real life. The valve is opening and closing. And here the green cells are sorted into the positive collection chamber. Microchip-based cell sorting is truly gentle. Cells do not get decompressed or charged, and only low pressure and mild shear forces are present. Another advantage is sorting in a closed and sterile cartridge is safe. There is no carryover, no contaminations, and no aerosols are being produced. If you are interested in high-speed sorting, we also offer our high-speed cartridge for fast sorting performance. Let's have a look at how the cells are arriving in the positive collection chamber. For this, an endoscope was inserted into the positive collection chamber, the cartridge was loaded, and the sorting was started. We can see here that the cells are easily flying into the positive collection chamber. Another advantage is that cells are strongly concentrated during the sorting process because the valve is only opening for the short time that the cell needs to be sorted. This can save you, for example, a centrifugation step. Sorting with the Max Quantido is aerosol-free. Here is a recent publication. The researchers used dragon green beads and a cyclic D impactor sampling cassette to check for aerosols in the environment. The Max Quantido was compared to a conventional droplet sorter. While the droplet sorter produced many aerosols during the process, the Max Quantido did not produce any aerosols, showing that it is possible to sort biohazardous material with the Max Quantido. A genomics workflow using the Max Quantido can look like the following. First, single-cell suspension has to be performed. For this, one could use the GentleMax system, or for example, cells can be pre-enriched using the Automax Pro. Then cells are sorted on the Max Quantido, and they are used for sequencing afterwards. Kui at the UCI is working on epithelial cells. He is interested in the diversity of the cells and therefore he is performing single-cell genomics. He is taking mammary glands from mice, doing single-cell suspension and sorting the cells via flow sorting. And afterwards he performs 10x genomics. Interestingly, he does a head-to-head comparison of the FACS droplet sorter and the Max Quantido. First, he observes that the cell types and states between the cells sorted on the Tido and the FACS droplet sorter are similar. However, when he takes a closer look at the data, he sees that the cells from the Max Quantido gave more gene reads, indicating a better sample quality. He is also looking at the stress response gene expression, and he sees that the cells sorted from the Max Quantido had a lower expression of stress response genes. Finally, he also performs a functional assay. Here he performs a sphere assay and he observes that the cells sorted with the Max Quantido form bigger and more spheres in culture, which is indicating a higher cell functionality. All in all, cell enrichment using the Max Quantido before sequencing improves the sample quality and reads while minimizing sequencing artifacts and costs. Last year there has been a webinar on exactly that topic. If you are interested, have a look at it. Nuclei sorting is getting more and more important in genomics workflows. Therefore, we also tested if this is possible on the Untitled. A confidential marker was used, together with DAPI, to stain for the nuclei. After sorting, a purity of above 95% was observed. I want to show you some recent publications using the MaxQuantilo prior to sequencing. One example from COVID-19 research is coming from Patrick Wilson and his colleagues. They are working on B-cell subsets. Another example from COVID-19 research is coming from Petra Bacher and her colleagues, and they are interested in the T-cell memory. A study working on biomarker identification is using oligodendrocyte progenitor cells. And there is an example from non-human primate research, where they sorted retinal cells. To summarize, microchip-based cell sorting is gentle for the cells. Sorting in a closed cartridge system offers the operator as well as the sample safety. One example for single-cell sequencing showed that sorting with a title prior to sequencing can improve the sample quality and the reads while minimizing artifacts and costs. Sorting of nuclei is also possible on the MaxQuantilo. With this, I want to thank you for your attention, and I want to hand over to Alexander.
Hello, my name is Alexander Jonsson, and I'm a PhD student in the Korsgren lab at the Department of Immunology, Genetics and Pathology at Uppsala University. Our research group is interested in the pathogenesis of diabetes, and my studies are focused on understanding the stromal cells of the human pancreas. I'd like to thank Miltenyi Biotec and Labut for arranging this webinar and giving me the opportunity to present my work. Today I'm going to talk about our recent application of the MaxQuantilo cell sorter to sort microvascular endothelial cells from human pancreatic samples. I'm going to start with a brief introduction to pancreatic microanatomy and highlight some advantages of using the MaxQuantilo cell sorter in our process. After that, I'll present a project I was involved in that was recently published. First, consider the human pancreas. Our group is interested mainly in the endocrine compartment of this organ due to its role in diabetes as the hormone-producing organ. However, the digestive enzyme-producing exocrine tissue actually constitutes about 99% of the pancreas. It consists of parenchymal cells, illustrated here in yellow, which is interspersed by a rich network of capillaries, seen here as red triangles. The hormone-producing part, the islets of Langerhans, make up just 1% of the organ and are randomly distributed throughout it. Similar to the exocrine tissue, they essentially consist of parenchymal cells with a rich capillary network. The average islet consists of about 1,500 cells. In the study I'm going to present later, we used the exocrine tissue compartment as sort of internal control to analyze the islet tissue. Now, the first step in our experiments is acquiring the tissue for analysis. This is done through a process called islet isolation, which is enzymatic breakdown of the entire organ into clusters of islets and of clusters of exocrine tissue. Pancreata are routinely received and processed for use in transplantation or research at our facility here at the Department of Immunology, Genetics and Pathology. If we are interested in the transcriptomes of the cells, we want to study the tissue as fast as possible, which often means starting our cell sorting at odd hours. And, well, this would make it very complicated to coordinate our cell sorting with some sort of core facility. So I'm going to show you an example of our pancreatic, microvascular, endothelial cell sorts. In this first figure, we actually see an unstained sample of islet cells, which highlights the quite high autofluorescence that is noticeable in our tissue. As you can see, there is a linear increase in fluorescence intensity for some of our cells, and we can see this in many different channels in our samples. Here's a demonstration of what our samples look like prior to sorting. After we have removed doublets through gating, we have about 1.5 to 2% of our cells being double positive for endothelial cell markers CD31 and CD44, which thus identifies these cells as microvascular endothelial cells. Then, after we sort the cells using the tidal cell sorter, we can reach a purity of about 90%. I think that the non-target cells that we see here down in the lower left quadrant are coincidental events that were sorted together with our target cells. It seems possible to reach a higher purity when we sort other cell types. So I think this is some inherent property of the endothelial cells that we're trying to sort that causes this contamination. So next, some things I quite like about the microquant tidal cell sorter and how we can use it in our workflow. It is a quite low maintenance and easily operated instrument. It does not require specially trained personnel and a core facility that maintains it all the time. We could implement it quite easily into our somewhat erratic workflow. Further, the cells can be kept at a consistently low temperature throughout the entire sorting process, both before, during, and after we've sorted the cells. This is important in order to minimize the metabolism in our cells prior to transcriptomic analysis. Additionally, the negatively sorted fraction is retained and can be easily used in downstream applications or be sorted. This is something we appreciate when we have our quite hard to get tissue material that we're trying to study. Additionally, the sorting forces themselves seem to be quite gentle for the cells, a very low fluidic pressure, no nozzle compression, no electric charge being applied to the cells, which would be the case in conventional flow cytometric cell sorting. This is quite nice, since this probably means that the sorting process itself has a minimal effect on the status of our sorted cells. So now I'm going to move on to talking about a recent project where we used the tidal cell sorter to sort microvascular endothelial cells. My colleagues and I did this work recently and had it published in the journal Scientific Reports. So why would we be interested in sorting endothelial cells? Well, the microvascular endothelial cells are actually a quite heterogeneous cell type that line the inner surfaces of capillaries. They are not simply a passive tubing, but they've been shown to differ between vascular beds in different organs and even within parts of the same vascular bed within an organ. When it comes to the pancreas, they also constitute an interface between the organ and the rest of the body, which means they might be implicated in physiology and disease, the interactions with nutrients, hormones, and inflammatory cells. They've also been shown to be able to actively signal with the islet cells, and they are known to be lost in islet culture and transplantation. Further, they are known to be morphologically altered in both type 1 and type 2 diabetes. Thus, we decided to investigate the gene-level properties of islet microvascular endothelial cells. So for a brief overview of our methodology, after organ donation, we get our isolated pancreatic tissue isolates with various contents of islets and exocrine tissue. We separately processed our islet samples with an islet content of 78% or higher, and our exocrine samples, which had an islet content of 0 to 4%. In order to be able to sort our cells, we first had to break down the larger islets and exocrine cell clusters acquired from isolation into single cell suspensions. This was done using the enzyme acutase. After this, we stained our cells using two markers, endothelial cells, CD31 and CD34, and one marker for leukocyte cells, CD45. We then sorted our CD31 and CD34 positive cells using the Taito cell sorter. Having done this, we took small aliquots of cells for quality control analysis. We assessed the purity through qPCR and flow cytometry on a separate instrument, and the majority of the cells were used in RNA-seq, and we then compared the data between these two compartments from our isolated pancreatic samples. And some results. So in total, we analyzed 8 organ donors with normal glycemia and 5 with impaired glucose metabolism, in order to be able to make additional comparisons between the healthy pancreas and the more type 2 diabetic-esque pancreatic phenotype. The impaired glucose metabolism group was quite heterogeneous, consisting of some donors with an isolated high HbA1c and some donors who also had treatment for type 2 diabetes. So here are some donor characteristics. To summarize this table, the two groups were highly similar for all clinical parameters, such as age, BMI, etc., but they differed statistically in the level of HbA1c, which is what we were going for when we chose these groups. So we analyzed the purity of our sorted samples in our aliquots. We used qPCR to analyze some genes that are typically expressed in islets, in exocrine tissue, or in endothelial cells. To summarize these data, we pooled the data from all our samples based on their sample origin. And here we can see that the endocrine genes glucagon and insulin are mainly detected in our islet samples prior to sorting, which is seen here in black in the top row. But there's also some detection in our sorted islet and endothelial cell samples, seen with the red part of these circle graphs. It seems that there's a bit more contamination in glucagon than with insulin. Then for our exocrine gene amylase, we can see that the majority of the signal was detected in our exocrine samples prior to sorting, with some level of contamination in our exocrine endothelial cell samples on a comparable level to the islet bulk samples. And finally, we see in the bottom row that the endothelial genes PKM1, which is the same as CD31, and von Willebrand factor are almost exclusively detected in our sorted endothelial cell samples. Next, for some post-sorting flow cytometric analysis. Here are two representative flow cytometric analysis of our sorted samples, showing that the purity of endothelial cells are roughly 90%. To further evaluate the purity of our sorted samples, we used multi-subject cellular deconvolution, MUSIC, a mathematical method for estimating the cellular composition of bulk RNA-seq samples by comparing the bulk transcriptomics data with single-cell RNA-seq data. Doing this, we also found that the model predicted that we had roughly 90% endothelial cells in our samples from both tissue compartments. And when we plotted this data versus the FACS data, we see that in general, these two measurements correlated well, with the exception of a few cases where the MUSIC estimate was higher than the FACS estimate seen here to the top, in the top left of the figure. So we have reasonably pure samples. Then do these samples differ at the gene expression level? And if so, how? First, did a principal component analysis to see if there was heterogeneity in our samples. And as we can see here, the samples cluster somewhat based on the tissue of origin, with our islet endothelial cell samples being in one part of the PCA and the exocrine samples in another. However, there was no evident separation of donors with impaired glucose metabolism or normoglycemia. This heterogeneity consisted of about 700 genes that were upregulated in our islet samples and 600 that were upregulated in the exocrine samples. In these figures, we can see the counts per million, that is a measurement of the level of transcription on the x-axis in the left figure versus the log fold change on the y-axis. And additionally, we can see how these genes distribute when we compare the log fold change with the log p-value in the right part of the figure. So then we analyzed these genes through gene set enrichment analysis, and we found that there are several gene sets that were upregulated in islets, seen here as red dots, and another bunch that are upregulated in the exocrine samples, seen here in blue dots. In this enrichment map, similar gene sets are clustered together in the yellow circles, which are then manually annotated based on their function. I've highlighted some interesting and highly similar clusters that are here in red that are upregulated in islets, and these sets are related to vascular development and endothelial development. Additionally, we can see some various gene sets that are upregulated in the rest of the figure in islets, including gene sets related to modifying the extracellular matrix and the regulation of various functions. I've also highlighted the upregulated gene sets for exocrine tissue, and these gene sets were related to biomolecular and ribosomal processing. And next we plotted the top upregulated gene sets. The go terms with enrichment in islet samples have a positive NES score as indicated on the x-axis. Again, I highlighted some of these gene sets in red. See that there's quite a lot of gene sets that have to do with vascular and endothelial development that are upregulated here in our islet samples. So what could this data mean? From our transcriptomic data, we can only speculate on this, of course, based on the earlier findings in the literature. There have been previous findings of increased replication in all pancreatic cell types in a subset of organ donors. This shows that turnover of the endocrine tissue is feasible, at least under some conditions. Additionally, there could be an extent of physiological islet cell turnover. Islet size is known to be quite heterogeneous, and the extent of cellular turnover within islets and whether smaller islets grow into larger ones and the factors determining the maximal size of an individual islet have not been determined. So, intra-islet angiogenesis and extracellular matrix remodeling from endothelial cells could be an important piece in this puzzle. Enablers of cellular turnover and remodeling. So for some conclusions then, we used the tidal cell sorter, provided a very convenient alternative for sorting primary microvascular endothelial cells at a high purity. This allowed us to create sort of atlas of genes that are likely expressed in the pancreatic microvasculature. And among these genes, we found higher levels of angiogenesis-related transcripts in our islet and microvascular endothelial cell samples. This is a transcriptomic data that is consistent with ongoing tissue remodeling that has been previously suggested in the literature. And in our exocrine microvascular samples, we found an upregulation in biomolecular catabolism. So we don't really know what this stands for, but it might be some contamination from the exocrine parenchymal cells. And we also did not find any unifying transcriptomic microvascular endothelial cell signature that differs between subjects with normal glycemia and subjects with impaired glucose metabolism. However, we had quite few subjects with impaired glucose metabolism, and they were also quite heterogeneous, with some not having a type 2 diabetes diagnosis and some having type 2 diagnosis treatment. So it's hard to tell what this stands for. There might be some more specific signature at more specific time points, the disease, for example. And that concludes my talk. Thank you very much for listening, and I'll be happy to answer any questions you might have during the live question and answers session.
Thank you, Alina and Alexander, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Our first question is, you mentioned it is possible to sort biohazardous material. Is it also possible to sort bacteria with the Tyto?
Thanks for this really interesting questions. Of course, sorting of bacteria is a really interesting topic. And actually, yes, this is possible. There have been E. coli sorted on the Tyto, and there has been a bioburden test also performed afterwards showing no cross-contaminations. It is also possible to sort yeast cells. Also, Plasmodium parasites have been sorted on the Tyto, and yeah, it was also possible to see the movement of these cells afterwards, which confirmed the viability of these cells. And interestingly, because due to the flexibility of the cartridge, it is also possible, for example, to sort anaerobe bacteria. For example, the buffer can be overlaid with an oil to keep out any oxygen. You can find all these data on our website if you are further interested in this topic.
Thank you, Alina. Our next question is, what are the challenges of studying isolated human pancreatic tissue?
Yeah, so I think there are some challenges both related to our single cell suspensions and also to the study design. So we have this high autofluorescence that I showed. That means that you need some quite bright stainings to be able to distinguish the target cells in your parenchymal cells. Then again, the islet isolation process, single cell dissociation, is a quite significant stressor for the cells. So they are probably quite susceptible to additional damage during further processing before analysis. There's also quite this extracellular matrix composition in pancreas between donors, which I think affects the number of cells we can get material and how well dispersed those cells are. And then the donors with diabetes, we can't really control our diabetic time points very well in those studies.
Thank you, Alexander. Our next question is, what kind of buffer do you recommend? What kind of buffer do you recommend for nuclei sorting?
Yeah, this is a really complex question. I cannot give you a black and white answer to this one, but what I can tell you is many customers are using homebrew buffers, which they were already used to employ for their sequencing experiments. These buffers are mostly based on PBS, and sometimes also stabilizers are used. For example, sucrose or BSA is also sometimes beneficial. But you have to be careful in case of a homebrew buffer. I would recommend to check the quality of your samples, especially if you have complicated samples which are, for example, debris rich. It is important always to have an eye on the quality. And yeah, as always in research, it really makes a lot of sense to determine the best buffer conditions, because this is of course crucial for the outcome. And yeah, based on your cell type and your sequencing application, it is an advantage to determine the best buffer conditions. You should also, of course, check if there are any substances that could maybe interfere with the sequencing application. Sometimes ions can be a problem, or also, for example, high concentrations of EDTA might be a problem. And yeah, so this is what I can say to this question. Thanks.
Thank you, Alina. Our next question here is, can you please tell me the procedure and time taken for the cell sorting?
Yeah, sure. So the majority of the time it takes is during the sample preparation. The single cell dissociation step and stainings and the washing steps and such take about 2 to 2.5 hours in our project. And then the sorting time itself is quite fast, maybe 10 to 15 minutes to prepare a cartridge. And then depending on what purity you're going for, you don't want to load too high concentration of cells. But if you're going for a high purity, you can sort about 1 million cells in 15 minutes. So I think it's quite fast, the sorting time.
Thank you, Alexander. And our next question here is, approximately how long does it take for the tissue digestion and preparation step?
Yeah, again, this is the step that takes the most time. So first we need to get the islets themselves from the intact organ donor pancreas. And that takes quite a long time, maybe 4 to 5 hours, and it's done at a separate facility. Then after that, we do the single cell digestion. That takes about, the digestion itself takes about 30 minutes, but you need to do the washing steps as well and the stainings and such. So 2 to 2.5 hours. So in total, 7 or 8 hours, I guess, for the tissue preparation.
Thank you so much, Alexander. And our next question, in context with sensitive cells, have neurons been sorted on the Taito?
Yeah, also a very interesting question. The neuroscience field is growing a lot, and we also have tested quite some applications in this context. Neurons and also neuronal cells, they have been sorted on the MaxQuant Taito to date. One example is, for example, neural stem cells. They were sorted from mice, and it was also possible to differentiate these cells into glial cells and also neurons. This showed, that the functionality of these sorted cells was good. And we also offer a sorting and analysis kit for this workflow. If you are interested, you can have a look on the webpage. There's a poster on this topic. In addition, yes, indeed, it was possible also to sort adult neurons from mice. Sorting neurons is, of course, very tricky. The cell type is really sensitive. You may know these cells do have long dendrites, and that's what makes them so sensitive and also tricky. However, it was possible to sort this cell type with a viability above 95%. And if you are interested, have a look on this on our webpage.
Thank you, Alina. Our next question: how many different cell populations can be sorted simultaneously from a sample?
So from one cartridge, you can sort one cell type, but it is then no problem, because the advantage of our system is that absolutely no cells do get lost. And that's why you can simply regain the cells from the negative chamber, and you can resort them again for any cell type you might be also looking at.
Thank you, Alina. And our next question here: what factors are central for high cell sorting purity?
Yeah, I think one of the central factors is that you don't have too high cell concentration due to the way the fluidic system works, and that you apply a threshold close to your cells when determining the sorting procedure. So that's highly important. You don't want to go above 1 million cells per milliliter. Then it also seems that there's some differences between different cell types, from my experience, that maybe some cells don't quite behave like a well-behaved single cell suspension. So when the endothelial cells, for example, seem to be able to reach a lower purity than some immune cell subsets within the pancreas.
Thank you, Alexander. And we've got another question in from the audience. What protocol do you use to dissociate your tissue? Enzymatic mechanical dissociation.
We used enzymatic dissociation with the commercial enzyme Accutase, with some added mechanical assistance with pipetting. Yeah.
Great, thank you so much. And here we've got another question. Can primary DCs be sorted on the Taito?
Yeah, primary dendritic cells are also a really sensitive cell type, also due to their shape. And indeed, there has been a recent publication on this topic. There were researchers from Italy, and they were sorting dendritic cells, interestingly, from healthy donors and also from COVID-19 patients. For this publication, downstream they used transcriptomics and sequencing, because they wanted to unravel the transcriptional response of the DC subset during the SARS-CoV-2 infection.
Thank you, Alina. And our next question. What are the biological implications of increased levels of vascular development transcripts?
Yeah, so I think it's our readout is a bit limited in our study. What we can see is that these transcripts are overrepresented in our islet samples, of course. So we can just speculate on what these increased numbers of transcripts could mean biologically. And there's some interesting work that's been done in the pancreas that shows there might be some increased islet cell turnover in a subset of organ donors, and such a phenomenon could mean that there will be an additional need for remodeling in the vasculature as well. So I think that's a quite interesting mechanism that should be investigated further.
Thank you, Alexander. What is the maximum flow rate?
Yeah, thanks for this question. So we can talk a bit here about the technology. As you might have learned from the talk, we do have two cartridges, a standard cartridge where you can run 4 milliliters per hour, and we do have a high-speed cartridge which runs at 8 milliliters per hour. With a high-speed cartridge, we take advantage of the physical feature inertial focusing, and it is possible to sort a maximum of 50 million cells per milliliter. You can load 10 milliliters in one cartridge. This means that you could load the cartridge with a maximum of 500 million cells. This makes for the high-speed cartridge up to 110,000 events per second. I think what Alexander can even go higher. This is really no problem. Thanks for this question.
Well, thank you so much for this answer. And we've got another question. Is there a size limitation for the cells that can be sorted?
The size limitation is 20 micrometers, but for cells in suspension. So as you know, cells that culture, they are usually bigger and flat, but in suspension, you can sort cells of 20 micrometers. If you're interested, if your cell type is compatible, please, you can also do a feasibility assay with our cartridge.
Thank you, Alina. Thank you so much, both Alina and Alexander, for your time today. Do you have any final comments for our audience before we go?
Maybe from my side, really thanks a lot for joining. I hope you enjoyed it. I'm really happy that we got the opportunity to present here our technology and also the research. If you are interested in the MAX-Contido technology, please contact us directly at Miltenyi at any time. You can also contact your local sales representative, of course. And I also want to say thank you to Alexander for presenting his research. So that was really interesting and I enjoyed it. Thanks for all.
Well, thank you again.
Thank you for listening, and I'd like to thank Miltenyi and Labruth for arranging this webinar.
I'm sorry, Alexander, if you have anything more to say, I didn't mean to interrupt.
Oh, sorry. Yeah, I would just like to thank Miltenyi and Labruth for arranging this webinar, and big thank you to you guys for listening in the audience and interesting questions. Thank you.
Well, and thank you again, both of you, Alina and Alexander, for your time today and for your important research. We would also like to thank Labruth and our sponsor, Miltenyi Biotec, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. Labruth will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.